students, this is Ms. Shim, and in this video we're going to be talking about enzymes. We're basically starting Unit 3. Unit 3 is all about enzymes, cellular respiration, and photosynthesis. In our textbook, this is Chapter 8, and Chapter 8 is all about enzymes. So we're going to be discussing chemical reactions, like all different kinds of chemical reactions, and enzymes and how enzymes speed up chemical reactions. And we do need to talk about the structure of enzymes. And as we learned previously, enzymes are proteins. We have to talk about protein structure one more time. Okay? All right, so let's talk about metabolism first. Metabolism is basically all of the chemical reactions inside an organism. And when we look at the cellular level, like where are some of these chemical reactions happening? And so how is the cell's function related to food chains? We're going to be learning all about food chains when we go over ecology, and you probably learned about food chains when you were, even in elementary school, where you have different trophic levels, and these trophic levels begin at the bottom with what we call producers. Producers are basically autotrophs, right? Autotrophs are organisms that automatically make their own food. They don't rely on other organisms as a source of energy. They rely on solar energy. They could rely on solar energy. They could rely on chemicals in the environment to create their own food. And so producers are kind of at the bottom of the trophic level, and they're going to be eaten by what we call herbivores. And herbivores are also called primary consumers, like the first consumers, because the first consumers get eaten by secondary consumers. And then secondary consumers are going to be eaten by tertiary consumers. So this is what we call a food chain. And these organisms are found on different levels. The bottom level being producers, and then you have the primary consumers, the secondary consumers, and then the tertiary consumers. But on a cellular level, like what's happening here with the producers, right? Producers are making their own food in the chloroplast, okay? And the chloroplast, remember, is a double membrane bound organelle where it receives and absorbs solar energy. And we're going to be talking about this in Chapter 10, all about photosynthesis. But in a two-step process, the organelle chloroplast is going to make food for the plant, and also indirectly food, or directly, food for the primary consumer when the consumer eats the plant material. Okay? And then when the primary consumer, secondary consumer, and tertiary consumer eat the organisms below it, they're going to break down the organic compounds in cellular respiration to make ATP, to make energy. And that's going to happen primarily in the mitochondria. Some of the food breakdown happens in the cytoplasm or the cytosol, but a lot of the ATP is going to be made in the mitochondria. I mean, we all know the mitochondria as being like the powerhouse of the cell, but that doesn't really explain the full story about what's going on. Majority of ATP is going to be made in what we call the electron transport chain, which is located in the inner membrane of the mitochondria. But more on that in Chapter 9, okay? And so in an ecosystem, there's going to be a flow of energy, and life is built on chemical reactions, and we begin with the sun. The solar energy is going to be absorbed by producers or plants, or what we call autotrophs. And what autotrophs do is they convert that solar energy into chemical energy. So they are transforming energy from one form to another. And that chemical energy is going to serve as a source of food for the primary consumers. So we're going to start with the sun. So solar energy is going to be absorbed by autotrophs and plants. And I apologize, but it got kind of cut off here, but solar energy is going to be converted into ATP and organic molecules. And we say organic molecules, the plants are going to take that solar energy and make sugars. And from those sugars and other nutrients from the soil, they're going to make proteins, they're going to make lipids, they're going to make nucleic acids. And then when herbivores like this giraffe eat the plant material, they're gaining that plant material and they're breaking it down. There's chemical reactions happening in the giraffe. They're taking that organic molecule and they're going to use it to make ATP, again, primarily in the mitochondria in cellular respiration. And then also they're going to use the organic molecules to make more organic molecules. Like the giraffe has to grow. It has to grow and develop. So it's going to make more carbohydrates, it's going to make more lipids, it's going to make more proteins, it's going to make nucleic acid as it grows. And then, unfortunately, this giraffe is going to get eaten by the secondary or tertiary consumers. And I'm kind of blocking it, and that's probably good because it's kind of a sad picture of a baby giraffe being eaten by this lion. But the lion is going to gain organic molecules from the giraffe. So when the giraffe makes organic molecules, it's food for the secondary and tertiary organisms, okay? So those organic molecules are going to be used to make ATP, and then also the lion has to grow and develop. So it's going to be making organic molecules itself. It's going to make carbohydrates, lipids, proteins, and nucleic acids. There's basically two different kinds of chemical reactions of life. There's two different kinds of chemical reactions that are happening right now in your cells. The first type of chemical reaction is where you are forming bonds. You're forming bonds between molecules. You're building like macromolecules. And when we're building and making bonds, there's a chemical reaction, and I hope you guys remember this. It's called dehydration synthesis, right? Where we're building bonds. You're synthesizing, making larger molecules, and these are what we call anabolic reactions, okay? So when we form bonds, we're building, and those are called anabolic reactions. The other type of chemical reaction that's happening right now, and it got cut off here, but little Fillmore is saying that's why they're called anabolic steroids, okay? So people that are like bodybuilders, they might take steroids, and they're called anabolic steroids because bodybuilders are building muscle, and muscle, to build it, you're forming bonds, okay? The other type of chemical reaction is breaking bonds. So when you eat food, right, you're going to break down those molecules to make ATP. And this is a reaction, not dehydration synthesis, but remember the other type of reaction? Hydrolysis, right? We're using water to break bonds. So not synthesis, but breaking or digesting, sorry. And they're not anabolic reactions, but they're catabolic reactions, okay? So anabolic means building and catabolic means breaking. One way to remember this is like catabolic, like a catastrophe, like a disaster, you're breaking something apart. Or I've heard some people say like, like a catapult can break down walls or such, okay? Or a cat can break things, okay? So that's what I've heard, catabolic versus anabolic, okay? So once again, this top reaction, this is going to be dehydration synthesis. We're building, in this case, like a disaccharide. So this is a anabolic reaction, okay? Whereas hydrolysis, we're breaking bonds, right? We're using water to break bonds, and that's called catabolic reaction. And this chapter, we're going to be discussing enzymes. And, for every chemical reaction in your body, there's going to be an enzyme that's responsible for each reaction, okay? There's going to be an enzyme specifically for dehydration synthesis, bringing two carbohydrates together to make a disaccharide. There's going to be an enzyme that's going to break the bond in hydrolysis, okay? I know that in class, remember, we talked about amylase. Amylase is an enzyme that breaks down starch. And so students put like a cracker in their mouth, and the amylase enzyme in your saliva starts breaking down the starch by hydrolysis. And so here is just a molecular structure of what's going on in dehydration synthesis, molecular example of what's going on. Dehydration synthesis, remember this is called a glycosidic linkage, when two monosaccharides are joined together by a dehydration synthesis reaction, and then in hydrolysis, water is going to break into OH and H, and that's how these two sugars or monosaccharides are going to be broken. and there's an enzyme. There's an enzyme for that, okay? Some chemical reactions release energy. So when we actually, the ones that break bonds, that hydrolysis reaction or catabolic reactions, they release energy. And we also call them exergonic. When we think about the amount of energy that's being released or energy being absorbed, in this chemical reaction where energy is released, we call it an exergonic reaction because energy is going out, okay? Energy is being lost. So when you break bonds and when you, by hydrolysis, you break down polymers or large biomolecules, then you're releasing energy, and that's a type of catabolic reaction, hydrolysis, okay? So digesting molecules, you're breaking it down into smaller molecules. There's less organization and there's actually less energy that's stored in those bonds because you have less bonds. Okay, so it's a lower energy state. And then some chemical reactions require an input of energy. So you have the release of energy in catabolic or exergonic reactions, but in the opposite sense, you have other chemical reaction where you're building bonds and you're storing energy. There's an input of energy, and that's going to be the anabolic reaction. And instead of exergonic, we call it endergonic. Okay, energy is going in. There's an input of energy. We're building molecules, and we're building molecules by dehydration synthesis. It's an anabolic reaction. I'm just going to move myself over. I feel like I'm taking up too much space, unnecessary space. Okay. So yeah, when we build molecules, we're making bonds. There's a lot more energy. There's energy being stored in those bonds, and so you have to input energy. Okay. So this is what a graph looks like, exergonic versus endergonic. In an exergonic reaction, energy is going to be released. So like here, you can see that the reactants, in a chemical reaction, the reactants is what we start with. And they are going to gain a certain amount of energy, but then the products at the end of the reaction, the products is going to have less energy. So if you look at the y-axis of our graph, the y-axis is basically the amount of energy, and the products have less energy than the reactants. The difference between the amount of energy in the reactants versus the products, that is the amount of energy that's actually released, okay? So like what you end up with has less energy than what you started with. And so where did that energy go? That energy was released, okay? That energy was released. And the energy being released, it could be represented in many different ways. It could be heat, okay? Like right now, we're giving off body heat due to cellular respiration, okay? Where a lot of the energy that's being released in the chemical reaction is lost as heat, as like body heat. So energy is released, like an example of that would be digestion versus, and it's the energy released, we can represent it as a negative amount of energy that's stored, and that stored energy is called delta G, okay? Or free energy, the change in free energy, that's the ability to do work, okay? So, and versus an endergonic reaction, that's the amount of energy that's absorbed or invested. It's going into the reactants. And so the reactants gain a certain amount of energy, and then the products have more energy than the reactants. So there's more products that is there's more energy in the products compared to the reactants. So there must have been energy supplied. Okay, and we call that positive delta G. So if you have more energy, positive delta G, those products have a lot of energy stored in their bonds. There's more energy stored in there. So it's more useful, I guess you could say. It becomes useful. There's more ability for that molecule to be used to work, okay? Because there's a lot more energy stored in there, okay? So organisms require energy to live. And where does that energy come from? It really comes from the coupling of exergonic reactions where energy is released with endergonic reactions where energy is needed. So over here we have energy being released because the bonds are being broken. So we digest a disaccharide, we break the bonds between the disaccharides, so you get two monosaccharides. Energy is released. And so in cellular respiration, when we break down sugars, that energy that's being released is going to be actually used to make ATP, okay? And that energy is going to be used for synthesis. Oops, let me just go back. So that energy is going to be used for synthesis, and you can end up with larger molecules. There's going to be input of energy to create larger molecules, to build bonds, to make bonds, okay? And so if it should say if an exergonic reaction is downhill, why don't they just happen quickly? So going back to this picture, what do we mean by downhill? So this is our exergonic reaction. Like, you know, if the product has less energy than the reactant, why can't it just turn quickly into the product? You know, why can't it just turn to the product right away? Okay. It needs the reactants, and you can see in this graph, the reactant actually needs to gain. It needs to gain, remember energy is on the y-axis, it actually needs to gain a certain amount of energy before it becomes a product. Those molecules, okay, those molecules in the chemical reaction, in the reactants, they need to gain enough energy so that the bonds can actually be broken and turn into the product, okay? So an example of this, of what we mean by this is hydrogen peroxide. Okay, so here is a bottle of hydrogen peroxide. Hydrogen peroxide is H2O2, and you know, they have an expiration date, okay? Hydrogen peroxide has an expiration date. It's usually a couple of years. And what happens eventually to hydrogen peroxide, it turns into water and oxygen gas, okay? So again, going back to this, like, why don't exergonic reactions just happen quickly? It's because covalent bonds are stable bonds, and those bonds, those molecules have to gain energy to break those bonds, okay? So hydrogen peroxide, it takes a few years for H2O2 to gain enough energy to become water and oxygen gas. And so that's one of the reasons why hydrogen peroxide is sold in brown bottles, these opaque brown bottles, because, you know, sunlight or just light even from a room, an overhead, the store, okay, the store light, those molecules can gain that solar energy, the light energy. And when they gain light energy, they're going to turn into water and oxygen gas. So storing them in opaque, in dark bottles prevents light from getting in, and it prevents the hydrogen peroxide from gaining energy and turning into the products quicker, okay? So, you know, for the sake of just keeping the H2O2 fresh, okay, they store and sell the hydrogen peroxide in brown bottles. There's a really pretty butterfly right outside my window. Okay. So hydrogen peroxide, okay, it turns into water and oxygen gas, okay, and it takes a few years for it to do that. There is an enzyme in our cells called catalase. Okay, it's catalase enzyme. It's a protein, and you can see here, this is what catalase actually looks like. And when we talk about proteins and protein structures, remember there's like 4 levels of protein structure. You have the primary, secondary, tertiary, quaternary. Well, catalase, it's functional when it's in a quaternary state. It's got 1, 2, 3, 4. It has 4 subunits that come together to make a functional catalase that breaks down hydrogen peroxide and turns into water and gas. So yeah, we do have hydrogen peroxide in our cells. When we break down fatty acids, you know, when we eat fats and lipids and we break them down, hydrogen peroxide is a byproduct. It's a waste product of lipid digestion, breaking down lipids. And hydrogen peroxide is, it's toxic, right? I mean, that's why hydrogen peroxide is sold at the stores, because it is antiseptic. It's toxic. It kills the bacteria, But then we have an enzyme called catalase that breaks it down into water and oxygen gas. So it doesn't harm us because we can get rid of it pretty much immediately, okay? It's an enzyme an enzyme catalase is going to break it down into two harmless molecules, water and oxygen gas. So that energy that hydrogen peroxide needs to gain in order to turn into water and oxygen gas, that hill that you saw in that graph, that's what we call activation energy. Activation energy. So activation energy is the initial input of energy that's required to break down the hydrogen peroxide into water and oxygen gas. Okay, so those molecules have to absorb. They have to absorb energy in order for those bonds to break. Okay, like if you have a log, a pile of wood, it's made out of cellulose, a polysaccharide. It stores a lot of energy, and you can burn it. You can, burn it, break those bonds with an input of energy. Okay, so think of that input of energy, that activation energy being like the energy source, like a flame. Okay, breaking down those bonds. So what's happening here is that enzymes are what we call catalysts. A catalyst is something that gets something going. So it's getting the chemical reaction going. And how does the enzyme do that? It lowers the activation energy. Okay, so the initial amount of energy needed is up there. Okay, so initially those reactants need to gain a lot of energy to become the products. And again, hydrogen peroxide could take years and years for it to gain energy and then turn into the products, right? That's the uncatalyzed reaction. But with catalase enzyme, the activation energy is much lower. The amount of energy that's required for the reactant to gain is much lower, so they can turn into the products a lot quicker. Okay, so that's the new activation energy with the catalyst. So that takes a lot less energy. The analogy that I like to use is that let's say that you're trying to save up enough money. So this is your bank account, whatever. You're trying to save enough energy to get a product, like to buy the new iPhone. Okay, to buy the new iPhone. It takes a long time to save up enough money or energy, okay? But with an enzyme, you don't have to save that much energy. You can get it a lot quicker. And that enzyme could be maybe a coupon, okay, a sale. Maybe the new iPhone is like there's a coupon, 90% saving, okay, 90% off. Then you can get it really quickly. You can get the product with less money. You don't have to save as much money. The enzyme reduces the amount of energy that the reactant needs to gain, okay? So enzymes are catalysts, okay? They're found in our cells. They're proteins that our cells make, right? Remember, ribosomes make our proteins. So what's a cell got to do to reduce activation energy, get help, chemical help from the enzymes? Okay, so enzymes are basically going to lower activation energy so that chemical reactions can happen faster, quicker inside of our cells. So let's talk about enzymes. They're catalysts, as we said, and they are made out of proteins. Some enzymes are made out of RNA. Okay, we'll talk about that in another chapter, but there are enzymes called ribozymes that synthesize RNA, that help synthesize RNA, and they act like enzymes, but they're actually made out of RNA. They facilitate how chemical reactions happen faster, quicker. They lower the activation energy. Enzymes are highly specific. I mentioned amylase. Amylase is the enzyme that breaks down starch, right? It breaks down starch. It doesn't break down anything else, okay? So enzymes are highly, highly specific, like pepsin. Pepsin helps break down protein. It doesn't break down starch, okay? They're highly specific. There's thousands of different enzymes in your cells, and they all catalyze, speed up a specific chemical reaction, okay? So I like to say that, you know, for whatever chemical reaction we're dealing with in our cells, there's an enzyme for that. There's an enzyme for every chemical reaction. There's an enzyme for that. It controls reactions of life, keeping you alive, okay? So let's talk a little bit about enzyme structure and some enzyme vocabulary. So this purple thing right here, this is an enzyme. It's a protein. It's a biological catalyst. And the enzyme, this is our reactant or substrate, okay? So our reactant in the chemical reaction, or we call it a substrate, okay? Reactant, substrate, same thing, okay? Those words are interchangeable. They will fit into the enzymes, what we call the active site, okay? So there's our substrate. And when it binds to the enzyme, it's called the enzyme substrate complex. They're together. It's a temporary association. They only stay together for a split second, and then you end up with a product, okay? The end result of the reaction is called the product. So like where the substrate fits, that's what we call the active site. That's the site of attachment for the catalyst, for the substrate or the reactant, okay? So in regards to catalase, let's say catalase looks like this, okay? Hydrogen peroxide fits in H2O2, and that H2O2 is, remember the chemical reaction? It's going to turn into water and oxygen gas. So catalase is going to break it apart and turn it into two things, hydrogen peroxide, and it's going to break it up into water and oxygen gas. And we're breaking bonds, right? We're breaking bonds. So this is a catabolic reaction. This is an exergonic reaction. Energy is going to be released. If there's a lot of hydrogen peroxide, sometimes you can actually feel the heat of it. But, you know, it's a pretty small molecule, so it doesn't release too much energy. But if there's a lot of hydrogen peroxide and a lot of catalase, a lot of chemical reactions, you can actually feel the energy being released in the form of heat, okay? So that's the active site. So we do need to talk about protein structure now because enzymes are proteins. So remember the 4 levels of protein structure. You've got the primary, secondary, tertiary, and quaternary. Let's first talk about the primary structure of protein. This is basically the amino acid sequence, right? So our example here, we've got serine, tyrosine, alanine, phenylalanine, valine, cysteine, tyrosine, aspartic acid, cysteine, and glycine. There's 10 of them. And so you have a chain of amino acids. These are all amino acids. So where they bond, right, those are called peptide bonds, right? A peptide bond is formed in a dehydration synthesis reaction between 2 amino acids, okay? Let me just quickly draw what an amino acid looks like. This time I'm going to draw the R group at the top because that's what it looks like according to this paper. Okay, so I've got the central carbon atom, the hydrogen atom. That's supposed to be an amine group, right? You've got the amine group. They drew their amine H on the top, but that's okay. Yeah, they did everything facing up or down, and then the R group is going to be on top, okay? So this is how they drew the amino acids. And then when it bonds with another amino acid, I'll just draw it like this, right? By dehydration synthesis. Okay, like this OH, this H, this OH, this H are going to come together to make H2O, water, and then this carbon and this nitrogen, they form a peptide bond. So that's what's happened here. You see this is a peptide bond between this amino acid and this amino acid. Here's another peptide bond, okay? Those are all peptide bonds between the 10 amino acids. So that's the primary structure of protein. It's just a sequence of amino acids. But we know that proteins, functional proteins, do not remain in this structure. They don't stay as just a chain, right? Remember, I forgot, right? This shows you the dehydration synthesis reaction. So this OH, this H, or this H, actually, I think it's that one, according to this diagram. So let me just erase this one. Okay, let me just erase that one. And those molecules are going to come together to make water. So H2O is going to come out, and there's our peptide bond. So this is what we call a dipeptide, dipeptide, di meaning two, right? And then peptide referring to the peptide bond that bonds these two amino acids together, okay? And then the secondary structure of protein, remember we said that it's like an alpha helix, right? The alpha helix. And then you also have the beta pleated sheets, a beta pleated sheet, okay? But how does it coil? How does it fold? It's a hydrogen bond. There's a hydrogen bond between a carboxyl group of one amino acid. So let me circle this amino acid right here. This is one amino acid, and then it's a hydrogen bond with another amino acid. Okay, so here is hold on, this is another amino acid right over here. So you've got a hydrogen bond between this negative oxygen. Remember, oxygens are going to be negatively charged because they're electronegative, and they're going to be attracted to this positive hydrogen. Okay, so you could see here that, see how this is our chain of amino acids, and now it's folded. It's folded. This is like the start of either an alpha helix or a beta pleated sheet. Okay, so the secondary structure of protein, you could have this folding, and then held together by these hydrogen bonds. Okay, or in this case, these are the hydrogen bonds between the carboxyl group and the amine group. Carboxyl group and amine group. So it's holding it in the secondary structure. So proteins are held in secondary structure by hydrogen bonds between amino acids, the carboxyl group, and the amine group. Okay? I like this diagram too because it shows you clearly the hydrogen bond between the amine group and the carboxyl. Amine group, carboxyl, amine group, carboxyl. Okay? The tertiary structure of protein is when you have the coiling and the folding of the secondary structure folded even further, okay? So yeah, you can see this protein, you've got the beta pleated sheath, and then you've got the alpha helix, but the strand or the chain of amino acids is like folded. How does it fold? Like, how does it fold? By several types of bonds, okay? There's 4 bonds that we're going to be dealing with here. You've got the hydrogen bonds, you've got the hydrophobic interactions, disulfide bridge, and then ionic bond, okay? I think on the next slide I can do a better explanation or a closer. Actually, let me go back here, okay? So this is an amino acid. So this is our R group, okay? This is the R group of one amino acid. So let me just draw, this is our CO, and then H, that's the nitrogen. This is our peptide bond, okay? H, C, H, oh, oops, and then this is the R group for that one. And, okay, so these are R groups that are sticking out. Okay, R groups that are part of those amino acids in our chain. Okay, so you could have like the coiling, you have the folding, okay, but then these are R groups that are coming out. And for the tertiary structure of protein, it's the R groups that are holding it in this tertiary structure that's causing our coil and the folding, the beta pleated sheet, to fold even further. Okay, so these R groups are the ones that are kind of sticking out, and then they're forming these type of bonds. So on the next slide, you can see, let's identify the 4 types of bonds or interactions. This first one, okay, so here's an R group. of one amino acid, and here's another R group of another amino acid, and they're forming a hydrogen bond. Okay, those are two hydrogen bonds that are formed between these two R groups. And then over here, you've got what we call a disulfide bridge. Here's one R group, here's another R group. And the thing about sulfur is that sulfur can actually form a covalent bond with other sulfur atoms in another R group. So this, let me just draw like as if this is our alpha helix, okay, our secondary structure, alpha helix. It's folding like this because of a disulfide bridge between two sulfurs that are close together in our amino acid chain, okay? And then what type of bond is over here? We've got a plus sign, we've got a negative sign. That's what we call an ionic bond, right? Bond of attraction of opposite charge. Bond of attraction of opposite charge. They cause this, this chain to fold, okay? And then this is what we call the hydrophobic interactions, right? What do you guys notice about these R groups? They're hydrocarbons, right? When you have hydrocarbons, you have no charge on them. They're hydrophobic, right? Hydrocarbons are going to be hydrophobic. They're going to be non-polar. And so this protein, remember there's water outside of our cells and water in our cells, and these enzymes are found in our cells, right? And so if there's water in our cells, these hydrophobic hydrocarbons, our side chains, they don't want to be facing outside. That's why you don't see a hydrocarbon out here. All the hydrocarbons are going to be found inside, tucked away from the water. There's like water all around this enzyme, okay? Just all around. And so those are the four different kind of interactions or bonds that happens, keeping our secondary structure in this tertiary form, okay? And then quaternary structure of protein is where you have two or more tertiary structures coming together, and they can form several types of bonds, keeping them together. And then this quaternary structure of protein is functional. Okay, two or more polypeptide chains join together to form a functional protein. Okay, and they're called polypeptide chains, right? Because you've got many peptide bonds that are linking the amino acids together. So here is a diagram showing you 1, 2, 3. There's 3 polypeptide chains, and they're together. You've got an ionic bond, you've got the disulfide bridge, you've got hydrophobic interactions. Oh, did I say hydrogen bond? Yeah, hydrogen bond, hydrogen bond, hydrogen bond, and then you've got the plus negative, that's an ionic bond. Okay, disulfide bridge, hydrophobic interactions, ionic bonds, and it's missing hydrogen bonds. But those are the 4 types of bonds or interactions that hold a functional protein more than 2 polypeptide chains in quaternary structure. So just an overview of the 4 levels of protein structure. You got the primary, which is a chain of amino acids. Secondary, alpha helix or beta pleated sheet. From there, the alpha helix or beta pleated sheet, they're going to fold further due to the 4 types of interactions: ionic bonds, hydrogen bonds, disulfide bridges, hydrophobic interactions. And then quaternary structure of protein is where you have 2 or more of these polypeptide chains coming together by those 4 types of interactions: hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bridges. So it's just, again, some important properties of enzymes is that they're reaction specific. Like you can't use amylase enzyme on protein, right? It's only going to break down starch. Each enzyme works with a specific substrate. Because the substrate and the enzyme, they fit together, right? They fit into the active site. It's like a lock and key. And we'll talk a little bit about why the lock and key analogy doesn't truly work, okay? But it is a fit. And when the enzyme and the substrate fit together, they form a temporary association where they stay together because of hydrogen bonds and ionic bonds that are happening between the enzyme and the substrate, okay? And another thing to know about enzymes is they're not consumed in the reaction. What does that mean? They actually can be used over and over again. So enzymes are not like a one use and throw away type of protein. You can actually use it thousands of times. You can use it many, many times. Enzymes can last, depending on the enzyme, they can last hours to days to weeks to maybe months, okay? They can be used over and over again. I mean, it's like your phone, right? Your cell phone. It's not like you get a call and then you're like, okay, that's one-time use, you got to throw it away. You can use it over and over again. A single enzyme can catalyze thousands or more reactions per second. Enzymes are unaffected by the reaction. And then also, the last thing we're going to talk about today is that enzymes can be affected by cellular conditions. So the environment that the enzyme is in can actually either inhibit them or can help them, okay? Any condition that affects protein structure can either inhibit them or help them, and that is like some environmental conditions or cellular conditions that we're talking about is like heat or temperature, pH, or salinity. Salinity is like the amount of salt in a solution. So like some examples of enzymes is sucrase, it breaks down sucrose. Proteases break down proteins. Lipases break down lipids. DNA polymerase, it builds DNA, okay? Because it's a polymerase, like it's making a polymer. It adds DNA nucleotides to a growing strand. Pepsin, I mentioned this before, it breaks down proteins, okay? It breaks down those polypeptide bonds, those peptide bonds, okay? And so enzymes are specific. Sorry, this meme kind of sucks. I drew it on, I think using, not even this pen, but I think I drew it using just my finger on the mouse pad. So it's not the greatest, okay? So this substrate doesn't fit. Substrates, enzymes are specific. It has to fit like a lock and key. They have to fit together, okay? So the lock and key model, it's a very simplistic model of enzyme action. The substrate is like a key that fits into the enzyme. It fits into the 3D structure of the enzyme's active site. And what holds it in there are these hydrogen bonds between the substrate and enzyme. Like a key fits into a lock. But that's not entirely true, okay? Because the induced fit model is a more accurate depiction of how enzymes work, okay? It's like a lock and key, but actually the enzyme changes shape, and the enzyme actually, when the substrate goes into the enzyme's active site, the enzyme hugs it. The enzyme gets really super close to the substrate and puts physical strain on the substrate, okay? The substrate binding to the enzyme causes this enzyme to change shape, and it leads to a tighter fit, okay? So that's what we mean by conformational change. Conformational change means like it's changing shape. And we'll talk about how that happens in a few minutes, okay? So what exactly does an enzyme do? They lower the activation energy, right? Excuse me. They lower the activation energy, the energy that's required, and they speed up chemical reactions. But we say that, but what does it actually mean? Oh yeah, like on a graph, it lowers the activation energy. But what does that mean? Like physically, like what does it look like, right? Not just a graph, but what does it physically look like when the substrate goes into the enzyme? And so enzymes, you know, they speed up chemical reactions of synthesis, of breaking down, right? In synthesis, what they do is that the substrate goes into the enzyme, and the enzyme brings those two molecules that are going to bond together closer. It brings the two substrates together close so they can form a bond. The active site orients the substrate in correct position for the reaction to happen, okay? It brings the substrate closer so they form a bond. Whereas with digestion or breaking down, the enzyme, when the substrate fits into the enzyme actually puts stress, physical stress on the bonds and breaks it, okay? It's like if I have this pencil, the pencil is my substrate and my hands are the enzyme, and it goes into the active site, the enzyme, it hugs it and it breaks the bonds, okay? It's like putting actual physical stress on the bonds. And so I really like this diagram or picture because you can physically see, you can see what's physically happening. So we have our enzyme here, okay, E for enzyme, and this is our S for substrate, and it's going to turn into the products. This is like breaking bonds. The enzyme hugs it, and you can see it here in this picture, puts strain on the substrate, the reactant, and breaks it, okay? Versus making bonds, the enzyme is going to bring all the products together and bring them in close, sorry, that's not the product, substrate together, and they're going to go into the active site, bring them super close so that the bonds can be made, and then you end up with the product, okay? So enzymes can speed up these reactions of either breaking bonds or making bonds, where the substrate goes into the active site. For breaking bonds, it puts physical strain on the bonds breaking it. Making bonds, it brings the substrates super close to each other so that the bonds can be made, so that the product can be made. And so the analogy is like where we're making bonds, the enzyme is like a matchmaker, right? It brings the two substrates together so they become one. It's sort of like the, at a wedding, it's like the pastor, the priest that brings the couple together, okay? Or sadly, in the other analogy where you're breaking bonds, it's like the divorce attorney that separates the two. Okay, so any questions? This is not live, so we're just going to move on. All right, now we're going to talk about the factors that affect enzymes. Okay, what sort of factors affect enzyme activity? Because we talked about how enzymes are proteins, and as we know, proteins, those bonds that hold proteins in, the three-dimensional shape, they're not necessarily the strongest bonds, like hydrogen bonds and ionic bonds. Those are not really that strong, okay? So let's talk about some factors that affect enzymes. There's enzyme concentration, substrate concentration, temperature, pH, salinity, activators, and inhibitors, okay? Those are the 7 factors that we'll be discussing in this video, okay? So with enzyme concentration, we've got enzyme concentration on the x-axis and then the reaction rate, okay? The reaction rate, the speed of the reaction on the y-axis. When we increase the enzyme concentration, like meaning when we add more and more enzymes into a cell, let's say, what do you think that's going to do to the reaction rate? Well, there's going to be a lot of enzymes around to either break bonds or make bonds, and so the speed of the reaction is going to speed up, okay? It's going to look like this. As the enzyme concentration increases, the reaction rate is going to speed up. But you'll notice that at a certain point, right over here at this enzyme concentration, it stays the same as over here. No matter how many more enzymes you add, it stays the same, okay? It stays the same. And why is that? Okay, so what's happening here is that as the enzyme concentration increases, so does the reaction rate. And I'm sorry that the graph got cut off at the bottom, but it's the same as the previous slide. So if these are enzymes, these little red guys are enzymes, and we keep on adding more and more enzymes, there's more enzymes to do work to break the bonds or make the bonds, okay? But then, and so if there's more enzymes, right, there's more around to either break the bonds or make the bonds to frequently collide with the substrate, okay? So if you got a lot of enzyme in the cell, there's a better chance of the substrate meeting up with the enzyme and going into the active site. So you guys, these are like, again, if my right hand is the enzyme and then my left hand is the substrate, if there's a lot of these guys, there's a higher chance of them colliding, meeting, and then the enzyme speeding up the reaction and making the product, okay? But again, why does the reaction rate level off after a certain point? Well, the limiting factor is the substrate, okay? We can add as many enzymes as we want, but then if the amount of substrate or the reactant stays the same, there's only so much work that the enzymes can do, right? If you can, if you have like 100 workers, enzymes, and you have 100, jobs for them to do, but then you add 100 more workers, they're going to eventually have nothing to do, right? Because they're going to run out of work to do. It's sort of like that, where the substrate becomes the limiting factor. If you just keep on increasing the amount of workers, at some point, you're going to run out of work to do. So not all enzyme molecules can find the substrate, because you're going to run out of work to do. Okay, let's talk about substrate concentration. So if we increase the amount of substrate reactant, there also it's going to look like this, where the reaction rate is going to increase, because if you increase the amount of substrate or reactant, there's a higher chance of collision with the enzyme. But then at a certain point, the rate just is the same. It levels off. And so why is that? Okay. So what's happening here is that the substrate concentration, as it increases, so does the reaction rate. Because the more substrate you have, which are these yellow guys, there's a higher chance of colliding with an enzyme. Okay. The reaction rate levels off. All enzymes are active, have active sites engaged. So you have all of these substrates kind of just in the cell, but all the enzymes are busy working. And so at this point, the limiting factor would be the enzymes. Okay, so we're not increasing the amount of enzymes, we've increased the amount of substrate, and all the enzymes are engaged, they're all doing their job, and that's why it's going to level off, because really the amount of enzymes is going to be the limiting factor. The enzymes are saturated. They're saturated. They're at the maximum rate of reaction. Okay, with temperature, the graph is going to look very different. This is what the graph is going to look like for enzyme activity in regards to temperature. So this is very cold, and then this is going to be very hot. And as temperature increases, as it's getting warmer and warmer and warmer, the reaction rate increases, and it's at its maximum point at a specific temperature, okay? So if this is like an enzyme we're talking about in our body temp, okay? If this is like 98 degrees, Fahrenheit, right? Then reaction rate is going to be at maximum, but then there's a steep drop off. As it gets beyond that point, it gets really hot, then the reaction rate decreases very steeply. 37 degrees Celsius is our body temperature. And so when we're talking about enzyme function and temperature, the graph is always going to look like this, okay? It's always going to look like this because all enzymes have a maximum or optimal temperature that they work at, an optimum temperature. Like for the enzymes that are in our body, they work best at 37 degrees Celsius or 98.6 degrees Fahrenheit. Because this is the point at which you're going to have the greatest number of molecular collisions. So, I mean, when it's cold, the molecules are moving very slowly. And when it's cold and molecules are moving very slowly, there's a less chance of them colliding. Whereas when they get warmer, they start to move around more. And as it gets warmer and warmer and warmer, they start to move around more, and there's a greater chance of collision. Human enzymes work best between 35 to 40, okay? So somewhere between here to here, okay? Our body temperature is 37 degrees Celsius. But beyond the optimum, you see a steep decline in enzyme activity. Why is that? Well, it's because that heat is going to disrupt the bonds that hold the enzyme in tertiary or quaternary structure. Okay, so remember the hydrogen bonds and the ionic bonds are pretty weak. And so when you add a lot of heat energy, it's going to break those bonds. So it's called denaturation. When an enzyme changes its three-dimensional shape, it's maybe tertiary or quaternary shape, then it's called denaturing. And the enzyme is going to change shape, and it's going to lose its three-dimensional shape, and it won't be able to do its job. It will not be able to take the substrate and turn it into the product. And that's why the chemical reaction rate declines steeply, because the enzyme has changed shape. So like if I draw an enzyme, maybe shaped like this, okay? But when we heat it up, and let's say that this is our active site, okay? That's the active site. And then when we heat it up beyond 40 degrees, then those bonds that are holding the enzyme in its shape are going to be broken. Maybe it's like now like this. Okay. Where's that active site? It's not there. And so the substrate can't bind and no product will be made. So the enzyme will not be able to speed up the chemical reaction. Okay. But what about cold? So how can we explain when it's cold? Why is the reaction rate so low? Well, again, right, when it's cold, the molecules are moving around really slowly. The substrate and enzyme are moving around slowly. There's less chance of collision, okay? Molecules move slower, decrease in collision between enzyme and substrate. So you guys, in the cold, enzymes are not denatured. Enzymes are only denatured beyond the optimum temperature, okay? So don't get that confused. Some students will say, oh, enzymes are denatured when it's too cold. Enzymes are denatured when it's too hot. No, they only get denatured when it's too hot. They don't get denatured when it's too cold. It's just that in the cold, the molecules are not moving fast enough. So this is what we mean by denaturing. If this is our three-dimensional shape of the enzyme, we denature it, it loses its shape, and the active site is lost, okay? Proteins can lose quaternary, tertiary, and secondary structures. When you cook an egg, you're changing the shape of the white protein, and because it changes shape and it turns opaque, okay? Now let's talk about enzymes and temperature. Now let's talk about enzymes and temperature in regards to enzymes that are found maybe in different environments. So we did talk about how enzymes in our body work best at 37 degrees. Well, you know, there are some organisms that are found in geysers, like really hot environments, and their enzymes work better at hot temperatures, okay? So our enzymes might work best at 37 degrees Celsius, a human enzyme, but in a geyser, that's supposed to be a geyser in the background, they work best at 70 degrees Celsius. So for us, our enzymes would be definitely denatured by the time it gets to 70, but for these bacteria that live in geysers, those bacteria, their enzymes actually work best at 70 degrees, okay? So they have this adaptation of being able to withstand very hot temperatures, and their enzymes work better at very, very hot temperatures. 70 degrees is about 158 degrees Fahrenheit. So how do ectotherms, how do ectotherms do it? Ectotherms are organisms that get heat from the outside. Ecto means outside, therm refers to heat. So they're not able to regulate their body temperature internally. I mean, sometimes you guys have heard them referred to as being cold-blooded. I mean, snakes are not like, we don't really say cold-blooded, we call them ectotherms, okay? They don't maintain body temperature internally like endotherms do. Mammals, okay, one characteristic of mammals is that we are endotherms, or warm-blooded, because we are able to maintain body temperature internally. And so a bobcat is our example in this diagram or graph. No matter what temperature it is outside, ambient temperature means the air temperature outside, no matter what temperature it is outside, a bobcat or endotherm like us, we're able to maintain stable body temperature. So even though it's 10 degrees Celsius outside, that would be very cold, they're able to maintain very stable body temperature at around maybe 38 degrees Celsius, no matter what temperature it is. Whereas ectotherms, their body temperature is pretty much the same as the ambient temperature. If it's 20 degrees outside, that's what they are, right? 20 degrees inside their bodies. And so they're ectotherms. So why are we talking about this right now? Well, ectotherms, they have enzymes that need to speed up chemical reactions so they can stay alive too. So an ectotherm, it wants to stay warm because, the warmth is going to speed up chemical reactions. The enzymes and substrates are going to move around faster. So oftentimes you guys will find in the morning, snakes or lizards sunbathing, okay? So they will go out into the sun and sunbathe to gain warmth so they can get their enzymes going, so they can get their metabolism up, so these chemical reactions can actually happen. Now let's talk about pH. So on our x-axis, we have pH 0 to 14, and then the rate of reaction on the y-axis. So what's going on here? Okay, what is going on here? Let me just put up everything here. So pepsin, so this green graph shows pepsin activity. It's an enzyme that's found in our stomach, and pepsin works best at a pH of like what? Like 3.5, between 2 and 3. So our stomach actually produces hydrochloric acid, HCl, which has a pH about 2. Okay, and so pepsin, our stomach is very acidic. Pepsin works best at a pH between pH 2 to 3. And that's perfect because that's the pH of our stomach, and that's where pepsin is found. And then a lot of the enzymes that are just found in our body, maybe in our bloodstream, work best at a pH of 7, which is neutral. Trypsin, which is found in our intestines is slightly basic. It's between pH of 8 and 9. Trypsin works best in a basic pH, okay? So what's happening? I mean, we just talked about optimum pH, right? But what's happening beyond the optimum pH and before we hit the optimum pH? So pH, any changes in pH, remember, by definition, an acid is a substance that adds H plus to a solution, whereas a base removes H plus from a solution. So when you when you have an acid, like let me just go back here, okay? When you have an acid like hydrochloric acid, there's a lot of H plus in the solution, and pepsin works best in a solution. It maintains its shape when there's a lot of H plus in the solution. Whereas trypsin works best, it's going to maintain its shape in a solution that has less H plus in the solution, okay? So if there are changes to the pH though, like if you put pepsin in a neutral solution or pepsin in a basic solution, it disrupts the bonds that hold it in its three-dimensional shape, okay? Because those H pluses are ions, and they can actually disrupt hydrogen bonds. They could disrupt ionic bonds, okay? So it's going to disrupt attractions between charged amino acids. It's going to affect the secondary and tertiary structures. It's going to denature the protein, okay? So the optimum pH for most human enzymes work best between 7 and 8. Anything beyond or less than is going to disrupt or denature the protein. Oh, so yeah, like pepsin works best at a pH between 2 to 3, and then trypsin works best at a pH of 8. Anything less than the optimum, anything beyond the optimum is going to denature, okay? So this is not like the temperature graph. With pH, it's denaturing on both sides of our optimum, okay? Whereas temperature, it's going to denature the enzyme beyond the optimum. But with pH, it's on both sides of the optimum is where denaturing happens, okay? Salinity has to do with salt concentration. So like salt is NaCl, right? And NaCl is an ionic substance created by an ionic bond because you've got the positive sodium ion, the negative chlorine ion. And if you have positive and negative charges, do you think that can interfere with ionic bonds and possibly hydrogen bonds? Yes, for sure, okay? So all enzymes work best at a specific salt concentration, okay? Optimum salt concentration. Anything less than or anything more than is going to disrupt the secondary or, it's going to disrupt the tertiary or secondary structure of enzymes, okay, of the proteins. So what's happening here is that salt concentration, changes in salinity, again, cations, you've got the Na plus, anion, Cl minus. If we remove or add too many of these guys, they're going to interact with the three-dimensional shape of the protein. It's going to disrupt the attraction between charged amino acids. Like it's going to disrupt the hydrogen bonds. It's going to disrupt the ionic bonds. And it's going to affect the secondary, tertiary structure of protein. It's going to denature the enzyme, okay? However, there are some enzymes, enzymes are intolerant of extreme salinity. Like too much salt can disrupt, right? The bonds that hold it in three-dimensional shape. And so there is the Dead Sea. Okay, on our planet we have the Dead Sea, and the Dead Sea is called dead for a reason, because it's so salty that it's so dense that actually when people go there, and it's like kind of a tourist attraction, when people go there and go in the Dead Sea, they float immediately, because you can't drown, because it's so dense that all that salt just lifts your body up. And there's nothing that can live there because there's so much salt. Okay, so let's talk about compounds that actually help enzymes. So far we talked about like denaturing enzymes, but what about some things that actually help enzymes? A lot of enzymes actually need help, and they need help from what we call activators. And there's a couple of types of activators. You have cofactors, and then you have coenzymes, okay? Cofactors and coenzymes help enzymes. And in fact, most of our enzymes do need cofactors or coenzymes to help them. The difference between cofactors and coenzymes, and you don't really need to know this, but cofactors are inorganic, whereas coenzymes are organic. That's like the main difference, okay? So like minerals like magnesium, calcium, potassium, iron, copper, those are inorganic. Okay, those are inorganic. Inorganic meaning there's no carbon associated with them. They're not attached to a carbon compound. Those are called cofactors. Whereas coenzymes are organic molecules that bind to the enzyme that help it. Okay, so a lot of vitamins like B3, those are coenzymes that bind to the enzyme that help it. Okay. B2, coenzyme A, okay? So those are types of activators that help the enzyme. Whereas inhibitors do the opposite, and we'll talk about the 4 types of inhibitors or inhibition. These are molecules that reduce enzyme activity. Competitive, non-competitive, irreversible, and then also feedback inhibition. So what is actually, let me just remove the examples. What is a competitive inhibitor? A competitive inhibitor is an inhibitor that competes for the active site. So like it's drawn here, but let me just redraw it. I've got an enzyme, and then you know how the substrate, it fits, S for substrate, it fits right into the active site. But a competitive inhibitor is a molecule that has the same type of shape, and it competes with the substrate. It's a little bit different, so I'm going to write it as CI for competitive inhibitor, and it competes with the substrate for the active site, okay? And it binds to the enzyme. Some competitive inhibitors are permanent, like they'll bind to the enzyme forever, and those guys, okay, those are called irreversible inhibitors, okay? But some competitive inhibitors just stay there temporarily, okay? But anyways, a competitive inhibitor, it competes with the substrate for the enzyme. You know, this might be somebody that you know at school that is, you know, competing for your love interest, okay? Penicillin is an example of a competitive inhibitor. It blocks the enzyme in bacteria so that bacteria cannot build a cell wall. Disulfiram or anti-abuse, this is an enzyme that treats chronic alcoholism, because alcohol is a toxin and you need to break it down. And so there's an enzyme that breaks it down. And if you don't have that enzyme, or if you have an inhibitor that will block the enzyme's use, then that toxin's going to stay in your system and you're going to feel super sick. You're going to feel really bad, okay? And so you're going to have a severe hangover, vomiting 5 to 10 minutes after drinking. So after drinking, you're just feeling really sick and you're just not going to want to drink. And so a competitive inhibitor, you can overcome the inhibitor by increasing the amount of substrate because they're competing. So if you increase the amount of substrate, then you can kind of block it out, okay? Okay, and a non-competitive inhibitor, let me go back here, it binds to the site other than the active site. So I'm going to draw my enzyme like this. Okay, and so you know how the substrate, Here's my enzyme, here's the substrate. The substrate binds to the active site. Well, with a non-competitive inhibitor, it doesn't bind to the active site, it binds somewhere else. It binds somewhere else. So this is going to be my NCI, my non-competitive inhibitor. And when it binds to a site other than the active site, it changes the shape. The enzyme changes, the active site changes shape. So there's my NCI. It changes the shape of the active site, and now the substrate can't fit. The substrate can't fit because the active site has changed shape. Okay. Non-competitive inhibitors are also called allosteric. Okay, allosteric inhibitors. Allo meaning other, and then steric refers to the other site. So there's somewhere else on the enzyme that the allosteric inhibitor binds to, and it ends up changing the shape of the active site so the substrate can't bind. Okay, so that's how an allosteric inhibitor or non-competitive inhibitor will inhibit the enzyme action. So it changes the shape of the enzyme so the active site is no longer a functional binding site. And there are some anti-cancer drugs that work as a non-competitive inhibitor. It stops DNA production in cancer cells. Cyanide, you guys all have heard of cyanide poisoning, and cyanide is poisonous because it's an irreversible inhibitor to cytochrome C, which is a necessary enzyme for ATP production. Cytochrome C is an enzyme in your mitochondria that helps produce ATP, and cyanide binds to it and it doesn't come off, and that's why it would lead to death. Okay. So these are the two main types of inhibitors. You have the competitive, right? It competes for the active site. Allosteric inhibitor, or the non-competitive, it binds somewhere else other than the active site, but it changes the shape of the active site so the substrate can't bind. And these are just two memes that I created where the substrate binds to the enzyme, but then the enzyme can be distracted and not bind to the substrate because of a competitive inhibitor. Okay? Whereas a non-competitive inhibitor binds to a site other than the active site. Sorry guys, these are pretty bad pictures. And the substrate's supposed to fit, but then when the non-competitive inhibitor binds to the enzyme, the active site changes shape and the substrate will no longer be able to bind. So again, I said this, how irreversible inhibition is where the inhibitor permanently binds to the enzyme. It permanently binds to the active site for a competitor. Allosteric, it binds to the allosteric site and it permanently changes the shape of the enzyme. And that's how a lot of nerve gases, insecticides work. Okay. So when we were talking about allosteric, right? It's a site other than the active site. So inhibitors, when they bind to somewhere else on the enzyme, it can change the shape of the active site and it causes the enzyme to be inactive because the substrate can no longer bind. Okay. Whereas there are some activators that bind somewhere else on the enzyme that causes the active site to be open so that the substrate can actually bind. Okay, so that's what I mean by allosteric regulation. It's like an activator binds to the enzyme, changes the shape of the enzyme so that it can receive the substrate. Whereas an inhibitor binds somewhere else, changes the shape of the active site so the substrate can't bind. So, the next couple of chapters, we're going to be talking about chemical reactions like photosynthesis, cellular respiration. It's a type of what we call metabolic pathway, where we start off with A and we get the product, a final product G. But there's usually there's a lot of steps involved, okay? It's kind of complicated. There's a lot of steps involved to get to the final product. Metabolic pathways are usually controlled in multiple steps. So B, C, D, E, F, those are what we call intermediates to finally get to the product. Okay, so there's a lot of chemical reactions of life. They're organized in pathways, divided into chemical reactions into many, many steps. And there's usually enzymes for every step. It's pretty amazing. Like, for example, glycolysis has 9 steps. Glycolysis is like the first step of cellular respiration. We'll be talking about that in the next video in chapter 9. But there's like 9 steps to glycolysis, where glucose splits into 2 molecules. And there's like 9 steps, and each step requires an enzyme. It's amazing how, remember I said there's an enzyme for that. There's an enzyme for every little step, okay? And so, yeah, enzymes are usually, they're grouped together. So like going back to that pathway, right? Usually the enzymes are together so that the intermediate can be transported to the next enzyme, which is going to speed up the reaction, okay? So usually when we talk about a metabolic pathway, the product can act as an inhibitor to one of the enzymes in the pathway. So this pathway is regulated. It's regulated because you don't want to make too much of a product. So your cells, it's pretty amazing how your cells do this. Your cells will cause the product to become an inhibitor to enzyme 1 so that we don't produce any more product G, okay? So your cells don't want to waste a lot of energy making the product G. You don't want to make too much. So the product G can oftentimes act as an inhibitor to enzyme 1. So this is a way that you guys, our cells, regulate the product. The product is going to be used by the next step in the pathway. Like that final product is an inhibitor of an earlier step, okay? It's going to be an allosteric inhibitor to enzyme 1. It's kind of cool how that works. It's what we call feedback inhibition. Meaning like it goes back and inhibits enzyme 1, so it stops the production of the final product. Because our cells do not want to make unnecessary product. You're going to accumulate the product, so that product acts as an inhibitor to enzyme 1, so you don't make too much of it, okay? So this is just an example of what we do in our cells. So like threonine, okay? Threonine is like a precursor to making a amino acid called isoleucine. And isoleucine is going to act as an inhibitor to the enzyme 1, that takes threonine and produces it into intermediate A, okay? So basically threonine turns into isoleucine in several steps. There's actually 1, 2, 3, 4, 5 enzymes involved here. But isoleucine, the final product, goes back, it binds to an allosteric site on enzyme 1 and prevents threonine from being able to turn into intermediate A, okay? So I believe that's it for our presentation. Yeah, that's it for our presentation. And so I hope that this video wasn't too long, but I hope it was helpful. And I hope that you learned a lot about metabolism and enzymes. Okay, all right, bye. Have a really good day. See you later.